factors affecting the enzyme activity. So there are many factors which are responsible for the activity related to the enzyme molecules. Let us take the first one as a effect of temperature. As we said that all enzymes except some of them which are made up of RNA molecules, those are proteins, and here as these are proteins, at a low temperature they remain inactive, but as the temperature increases the activity also increases, and the peak level of the activity reaches at an optimum range of temperature, and beyond that optimum range, that is at high temperatures, the activity decreases because of denaturation of the protein nature of the enzymes, that is destruction of the enzymes will take place at high temperatures. So like if you draw a graph showing that rate of reaction to that of activity of the enzyme. So here initially the activity will increases, and at one point you will reach the peak level, and beyond that here it will decreases. So this is the temperature. So at 0 degrees temperature it will be gradually less, and beyond that, as the temperature is going on increasing, the rate of reaction will also increase. At one particular temperature level it is reaching the maximum, and this is called as a optimum temperature. And beyond the optimum temperature, if the temperature is increasing, what is happening to the rate of reaction? It will be coming down. So likewise this can be shown in an graph related to the activity of the enzymes at a particular temperature. The optimum temperature for most of the enzymes is 25 to 45 degrees centigrade, and here there is an temperature coefficient. Here for every 10 degrees temperature, what happens? There will be a change in the rate of reaction. So this is so temperature coefficient. So here for every rise of 10 degrees of centigrade of temperature, the rate of reaction will, or activity will doubles. This rise in the rate of reaction of activity of the enzymes for every 10 degrees of temperature is called as a temperature coefficient. Then pH. So here majority of the enzymes are active at neutral pH, that is 7, but depending upon their nature, so some are active at acidic pH and some are active at alkaline pH also, but majority of them are active at the neutral pH, that is 7. So let us take some of the examples which are active at acidic, neutral and alkaline pH, like pepsin, which is active at 2 pH 2, and lipase, which is active at 4 to 5 pH, and amylase, which is active at neutral pH, that is 7. Then catalase is also active at neutral pH, that is 7, and trypsin is the one which is active at little alkaline pH, that is 8, and lipase is one other example which is also active at 8 pH. Likewise, some of them are active at acidic pH, majority of them are active at neutral pH, and some of them are active at the alkaline pH also. So, next factor is the concentration of the substrate. So, there is a direct relationship between the concentration of the substrate with that of the catalytic ability or rate of reaction, because here as the substrates go on increases, the catalytic ability will increases. But here if more number of substrates are available beyond the limit of the enzyme, that is if all that catalytic sites have been occupied by the substrate molecules in the enzyme, now further there is no space for the enzyme to provide active sites for the remaining substrate molecules. So, at that time the variation can be seen. That is what the concentration of the substrate is also responsible for the one of the factor which is responsible for the catalytic ability of the enzymes. So, here if more catalytic sites are provided, then the substrates are participating in the reaction and the products will be formed. So, here that can be shown in the form of a graph where the velocity of the reaction and the substrate concentration. As the substrate concentration is increasing, then also the rate of reaction will remain constant. So, initially what is happening, when the substrate concentration is increasing, the velocity, the rate of reaction will also increasing, but beyond that limit, that is when all the catalytic sites have been occupied by the substrate molecules, now there is no further catalytic sites on the enzyme to provide for the excess substrates which are present. So, the substrates are increasing, then also what is happening to the rate of reaction will become constant. So, this is called as a maximum velocity or maximum reach, whereas here this is called as Vmax. So, here this is a velocity V, and this Vmax is called as Michaelis constant. This is called as a Michaelis-Menten constant. So, here substrate concentration required to cause half the maximum reaction rate is termed as Km, that is Michaelis-Menten constant. So, here this is a graph showing the Km that is Michaelis-Menten constant. So, likewise the concentration of the substrate is also been one of the factor useful for the specific activity or the rate of reaction by the enzymes.